device that we could manufacture very reasonably so that we could service basically the whole world.
Soil health it's a healthy microbial ecosystem, and we give you a glimpse into that very quickly. There's nothing like that.
For over 50 years, we've known that microbes are the best indicator of soil health. Over 2 million articles have reported this, and they've been using the test chloroform fumigation assay, which in a laboratory costs $655. But there's been 2 million reports showing that it correlates with soil health, with soil nitrogen, with soil carbon. The other method people have used to look at that is microscopy. The problem with microscopy is that microscopy only detects about 1/10,000th of what we can detect, because we've gone in and developed a method of extracting the microbes from the soil. We can show that our sample is more than 95% pure microbes. So we'll show you a picture of the soil that microscopists are using to try and calculate the number of microbes, and a picture of what we're using. And we're using 3 drops on our membrane. We're detecting these microbes by their pigmentation.
So, Judy, how do you extract the microbes from the soil? You said that the traditional microscopy takes a smear of wet soil on a microscope slide, and then how did you figure out how to, concentrate the microbes away from the mineral parts of the soil?
So what we've done is we've developed an extraction fluid that contains different salts, and these salts, together with a careful whisking, they separate the microbes from the soil. But the salt that we have, our extraction powder, also manages to precipitate the soil, so that after we've extracted the microbes from the soil, we now then precipitate the soil to the bottom.
So, Judy, now that you've got your extracted microbes and we placed them on the test card, what is the function of the test card? Why did you design it that way?
Once we've separated the microbes from the soil, we can detect them either by spectrophotometry.
Okay.
That's not handy for the field test.
What, a microscope?
No, spectrophotometer is not handy either. Too, right. So we put 3 drops on a membrane. So we carefully chose a membrane that doesn't bind any of the common pigments you might see just floating around in soil. It only collects microbes on the surface of this membrane.
All right. So it filters, kind of like the liquid goes away.
The liquid goes away, and if there's a color in the liquid also goes away.
All right.
So if people have used humic acid.
Well, I did the test with molasses, and it doesn't change anything. And I put a lot of molasses in.
Yeah.
So it works.
Different things that people put in these things don't affect the assay. Now the microbes are trapped on the surface of the membrane, and the color that it gives to the membrane can be compared to a grayscale.
Okay, right.
Which tells you that the intensity of the color, not the color itself, but the intensity of the color correlates with the quantity of microbes that we have there.
Right, just like in a colorimeter or spectrophotometer.
Right.
It's the intensity of the color is linearly related to the concentration of microbes.
Right.
Right.
Actually, I came up with this grayscale in the beginning because it's a quilting secret.
Really?
Quilters.
All right.
Okay. Want to make sure that they not only have different colors, but they have different intensity of color. So they're not just measuring color, they're looking at intensity of color.
Okay.
And for that, they use a red filter,
All right.
and they use a gray scale.
So it turns it monochromatic. Basically.
It's monochromatic,
Right.
okay? And that version of it that we have now that still can be read monochromatically
Right.
is being used by the Amish.
Right, because they don't use cell phones.
Because they don't use cell phones, right?
Well, so when she came up with this, you know, I was thinking, and I was trying to do this monochromatic measurement, right? So we basically put a piece of red cellophane over a grayscale and the sample window, and we had to judge, you know, how dark is that thing, and you know, it's Judy and three men sitting around the table. None of the three men could do it because we can't. Our color detection is terrible. All right, so I think that's why we decided to go with the cell phone app, but I based it all off of Judy's quilting secret. And that's how we got it. So.
Yes, and then the New York Times confirmed that 25% of men are basically colorblind.
Or just color stupid. I don't know what it is.
So a color test really doesn't work for a lot of people in this world.
Yeah. And plus, you know, if we go to the app, then you know it's you can store a lot more data that way, right? I mean, you do the analysis of the test card. And then you can keep all these details about the soil or the compost or whatever you're looking at and start to keep records and look at changes over time. It's just a nice little bookkeeping method, having it on an app. So.
People like a number. You can compare a number. It's very hard to compare something that's just a choice, a quality choice, or a choice like that. And it's hard to do scientifically, too, if you don't have numbers.
Right.
So the app is, I think, interestingly, it was enabled by the fact that we tried to make a very cost-effective device for the membrane. In order to make it cost-effective, we had to make it lateral flow. Almost all of the medical devices do vertical flow.
Yeah.
And a vertical flow gives a lot of technical problems. You have to put different types of membranes together. Mm-hmm. They have to, and then you have to use a clamshell type device that presses it down. Mm-hmm. And if the pressure isn't right, because I've worked with a lot of them, you get a lot of seepage around the outside. Okay, so, and these devices are very expensive. Yeah. And they have to be, it's very expensive to assemble them also. Right. So we needed a device that we could manufacture very reasonably so that we could service basically the whole world. And then the flatness of our device meant all the other devices that are vertical have a rim around the area where the membrane is. Yeah,
it's literally called like a sample well.
It's a sample well.
So, and you look down a well, can you see the bottom? No, because it's casting a shadow. So now that we're flat, you don't cast shadows, we can read the color.
And the other thing that is the lateral flow is actually more rapid than a vertical flow. Right. So the lateral flow allows us to put the sample on much more rapidly than you can when you're using a vertical flow device. So Brady, why has it taken you so long to optimize this test? And I won't overlook the fact that right now, you are the only one in the world who's been able to do this.
All right. But it did take a while.
Why did it take a long time?
All right. Well, so cell phones, they have a camera, right? But all the manufacturers have different software. So the image you're looking at isn't raw camera like you would on even a DSLR. That's software enhanced too. So you have to overcome the differences between phones. And we do that with the grayscale, but also we have a backing of a monochromatic color that sort of tricks all the phones to be in the same range in their software, in their white balance or whatever. And then there's another problem of different color temperatures. So when you're out in the sun or on a cloudy day, or you're in the shade on a sunny day, that light is extremely blue. And when you're sitting in your living room and you've got a little 60-watt light bulb there, that light is yellow-red. And if you're at the office and you've got your fluorescent lights, that light, at least for cell phone cameras, turns out to be white, where red, green, and blue are all equal. So we had to figure out a way to account for differences in cell phones and differences in ambient lighting conditions. One of the ways was to turn the flash on the light. So that works. That helped a lot. And the second thing is to have the same color background that fills the screen of the image when you take the measurement. And then, you know, the other technical aspect of the app is feature detection. So we have to be able to know that, you know, how does the app know that it's actually looking at a test card and that there's microbes in the middle of that test card? And so that took a little while. Well, like I said, it was just a lot of trial and error and testing, and Jeff and I were literally running from light source to light source with 5 or 6 different phones. We'd have a test card, and I'd hold 3, he'd hold 4 or whatever, and we'd image the exact same thing 7 times in outdoors, in the shade, in the sun, and in 2 different lights indoors. And when I got an algorithm that made all those numbers the same, I was good to go. That was it. That's why it took so long, because it's a long way to different light sources.
It's a lot. I've followed it, so I know why it took so long.
Right.
But basically, the secret then is the algorithms you have that compensate for the different light colors,
right,
for the different light intensity,
right,
and for the different phones.
Right.
And that's patented.
Right, correct.
So Brady, one of the things you noticed when you were working on this, and we've been working for a while on getting a fungal to bacterial ratio, was you noticed that you could discern the difference between these two. And would you like to talk a little bit about that discovery and how we implemented that?
Right. Okay. So we were using microscopy to validate the test. So we take we take 3 drops. You know, one little tiny drop, 14 microliters, and we put that on a microscope, and I would image 10 fields on that, and we'd have 3 cover slips. So that's 30 images of the microbial extract. And I wrote some software that could actually pick out the particles. And, you know, Judy's the microbiologist. And I said, do you think there's a size way of differentiating from bacteria and fungi? And she's like, well, the largest soil bacterium is going to be 2 by 5 microns. So I said, okay. So we just made a hard threshold of 10 microns squared. And I said, anything that's smaller than that, we're going to call a bacteria. Anything that's larger than that, we're going to call a fungi. And what we found was that when we got a fungal to bacterial ratio on the microscope, I could see a change in the color. There was a correlation between the color of the sample and that ratio that we found microscopically, which blew us away. I mean, that was not expected. We were looking at all these different methods of trying to differentiate them with stains and so on and so forth, and it turns out they're naturally very, very slightly different colored. Not a lot, very slightly. And the fact that the cell phone can pick that up is pretty cool. And so we got that, and then that's another patent.
Could you also tell me, like, one of the big advantages of the system you have is that the data is stored on the cloud.
Right, right.
And could you explain, like, how it's stored and how people can access and are accessing that information, pulling it together?
Right. It's stored on the phone and in the cloud. And when we first wrote the app, before the project management feature came along, there were maybe 10 different fields that went along with each sample, like, crop quality, crop type, soil class, those generic stuff. And then we realized that people who are using this test a lot, maybe they certainly know more about soil testing and what goes on in farming than I do. So I said, why don't we just open this up? We'll open it up and people can make their own fields, and now you can have as many columns of data, so to speak, as you want. And it's totally adjustable by the user. And the other benefit of having everything on the cloud is that, let's say you're head of a team and you've made a project and everybody's sampling different areas. But you can go in as the head of that project. If all the samples belong to a project, you can download it all. So you don't have to look at this person's phone and that person's phone. And they just all are there, and then you can analyze.
Well, if you're running like 3 or 4 different projects too, you can make each one a separate project.
Correct.
So that you put your data together on Farm A
exactly.
in one project, and
right.
Farm B another, and Farm C another.
Right. We've got a user that I'm in contact with a lot who's got, I think, 20 different projects going in. It's for every property that he manages has got its own project. So.
And that's so important because sometimes I get people who say, this field has been organic and this field's not organic. And I'm getting the same reading in both, okay? And then I have this other field that went organic, and it's looking really good. So why aren't both organic properties doing the same? But organic only really works if you're using the right organic.
Okay.
You don't necessarily improve your soil just by saying I didn't use a chemical.
Right.
You have to do something to improve poor soil.
Right. Right.
So this allows you to see if you're actually making that kind of an improvement.
Right, and it's exactly with the project management, having the data, you could do over time and over space. With an inexpensive test like this, $5 to $10 compared to $650, you can sample a lot more per acre. You can get more detailed understanding of your crops and the land that they're grown on. And then, as you do amendments, as you add certain organic type fertilizers, then you can see over year or season over season that your microbiology is increasing, your soil health is increasing.
So it's important that the microbiology increases because there's such a high correlation, CFE shows that, between like the water holding capacity of the soil.
Right.
So it makes your crop more resistant to droughts, and actually it makes your crop more resistant to water, to excess water and stuff like that. And the amount of microbes in the soil prevents erosion, so you're preventing erosion.
Right, because don't they?
Because it increases the texture of the soil, because the glues that they make that stick them to the soil remain after they die, and they cause the soil to be clumpy.
Okay.
And that clumpiness also allows it to hold more oxygen. Okay, so you want aeration. Compaction means low amount of microbes.
Okay.
So you can, improve compaction. You can improve water holding capacity. Okay. You can improve the amount of carbon in your soil, because the stored carbon in your soil is the bodies of dead microbes.
When you run our test, you're looking at a snapshot of the microbes there. It's not like that same bug, that same microbe is going to live the whole season. They're constantly turn over. They're food for other predators in the ecosystem, in the microbial ecosystem. And so, but that turnover, you can get a snapshot. Okay, I've got a lot of microbes there. That means that the whole ecosystem is being fed, right? And when you feed the ecosystem, then it goes all the way up to earthworms. Now, we don't have direct data on correlation of microbes with earthworms. At least I don't know of any. But, that's what soil health is. It's a healthy microbial ecosystem. And we give you a glimpse into that very quickly. Very quickly. There's nothing like it.